Hi, everyone. I'm Sarah Dudgeon. I am currently co-leading this awesome thing we have just recently started up called the Pathology Innovations Collaborative Community Trainee Group. That's the PICCTG for short. And this trainee group was started under the FDA's newest collaborative community. Collaborative communities are just opportunities for scientists that are working in all kinds of fields to build up these like interest groups in which the FDA can be a part of the group. They can join the conversation. So the FDA is a member of or a listening and occasionally a voice in the PICC. And within that collaborative community, we've started this trainee group. Also within that collaborative community are a lot of other projects that are running. And the idea is maybe this trainee group could be used as a tool for all of the trainees who are here today. You can use this trainee group to promote your research, to find new collaborators, to get to know experts like Lakshman, our guest today, and so many others. So we want to offer that kind of support and knowledge base, voice for all of you to, you know, start your careers or continue them, bolster them, etc., in regulatory sciences. So, Julia, I'm going to ask that you throw some links in the chat. What else do I have? Okay, I'm gonna. So I'm Sarah Dudgeon. Like I said, I'm co-leading this group with Julia and Kingsley right now, but we want to share the love. So if you guys are interested in stepping up into a leadership role, please let us know. I first met Lakshman when we were first starting up this Pathology Innovations Collaborative Community. After I finished my master's in public health, I started working for the FDA in CDRH in OCEL, which is their research group. So I started to get an understanding of what's behind the FDA. It's not just the logo, it's not the black box that maybe turns something down or maybe slaps on too many regulations. It's people, it is scientists, community members, policy. There are so many humans and so much thought that goes into the FDA, and it is complex. It's hard to understand. And particularly for trainees, you know, there's so much reading you can do, but one of the best things that I found for me to learn about the group that I was working for was really to speak to the experts. And one of those experts is our guest today, Dr. Lakshman Ramamurthy. He worked for the FDA for a long, long time. And now he's working at GlaxoSmithKline, which is a pharma group, and he is representing their diagnostic space and their digital health space. And so he's still interacting with the FDA to this day. He wrote an amazing book chapter, which we have linked in the registration to this event. But maybe Julia, if you can throw that, that link to his book chapter again into the chat. I found it super helpful. I know everyone else that I've spoken to about it has also found it super helpful. So just before we get started and I pass the mic over to Lakshman, I just want to cover a few ground rules today. Hopefully it is clear to you all. It is super informal. It is a time to kick back and relax and listen to some great information, meet some peers, and ask your burning questions. On top of that, there's no slides today. Obviously, we do want to be sensitive to any information that Lakshman obviously cannot divulge, you know, anything covered by not yet covered by patents and whatnot. So with that, I'd like to say throw your questions in the chat as we go. There will be some time at the end to ask them, but Julia is going to help moderate that chat, And Lakshman, I'd like to pass it over to you.
Hey, Sarah, thanks for inviting me. It's always fun to be around smart young people who are the next generation. So this is exciting. Hello. So I can introduce myself briefly in my career, and then would that work for you, Sarah? And then you can jump into stuff you've prepared. I know you're prepared for this session. So yes, I am at GlaxoSmithKline. I head up a precision medicine digital health regulatory group, where essentially I have a small team that works with me, and what we do is, as you know, precision oncology is very much the art of the day now, where ever since the genome was mapped and sequenced, we've had a much better shot at trying to arrest cancer in its march. So there have been a series of precision targeted therapies that have been approved. And so increasingly, big pharma as well as biotech are doing clinical trials that include in them biomarkers to try and either select for patients a priori because they know the mechanism of action, or on the other hand, they also can use them to stratify and see if patients do better in response with the biomarker versus without. So my group therefore works very closely with what's called the experimental medicines or biomarker translational group. We work closely with the regulatory on the drug side, and we also work very closely with the outside diagnostic partners. So it's a very matrix function. It's almost like this Zoom gallery view. We're all together and we all have to interact with each other. But prior to coming to GSK, which was about 2 years ago now, I was at Foundation Medicine, which is where I had the fortune to see people who should not be on Mount Rushmore, like John Iafredi and Joe Leonards. I make that, it's an inside joke. But I worked at Foundation Medicine as global regulatory lead, where I was part of a team, a great team indeed, that took our first NGS panel-based oncology tests through the FDA and also through Center for Medicare and Medicaid Services. And I'll go into that a little bit later on how these various agencies interact and intersect. And prior to working at Foundation, I used to be a consultant, which is when I first met John Iafredi. John Iafredi used to be part of a company called ArcherDX. He was a founder and advisor, and he was my client when I was a consultant. And then before that, I worked at the FDA for 6 years. So I'll wait for Sarah's list of questions on getting into more detail on the role of the FDA and what I did. But I don't know what you had in mind next. Should we go to the chapter next, or you want to take it back now and then you can send it back to me?
I would love for you to give us all kind of an overview, so we're all starting on the same page. Your chapter covered Class 1, Class 2, Class 3, Special 510K, abbreviated 510K of all of the standard pathway options, and then it also covered non-standard HUD and HDE, de novos, real-world evidence, and then LDTs, which I know we kind of want to hit specifically when we start talking about policy, because you do have quite a bit of experience there. So love to pick your brain. But can we start with just an overview of the standard pathways?
Sure, yeah. So for those of you who are interested in knowing how FDA came about, it's 1932, I believe, when first FDA was founded, and based because of safety issues on a drug called thalidomide, which caused children to have terrible defects caused by thalidomide. And that's when the country realized as one that we need to regulate drugs and regulate things that patients or people imbibe. And so the Food, Drug, and Cosmetic Act was passed. And the next milestone that concerns me in this talk is in 1976, is when there was a new regulation on medical device amendments. There was an amendment to the existing Food, Drug, and Cosmetic Act. And that 76 is then the birth of the medical device regulation. And it's almost, well, 40 years now, past 40 years, 45 years, that we've had the medical device amendment in place. So when the 1976 medical device amendment was passed, the idea is how do you regulate devices? And this is something I am a bit of an FDA geek, I'll confess to you early on, is I think, and this is, while this may seem trivial after I explained it, it wasn't trivial at first. People typically classified in other countries, that I've been to other global agencies, where they've classified them based on technology, saying, let's do all the next gen sequencing here, let's do all the PCR here, things like that, based on technology. But you can see that that can get cumbersome because technology keeps growing and we keep adding more tools. So the agency has a very simple approach to classification, and it's called risk-based, because it doesn't matter what technology or what device or what diagnostic or what implantable device it is, what is the risk to the patient? And the risk to the patient is also, therefore, then covered by the intended use. What do you intend to do with the device? When I give these sort of lectures or workshops, I explain the example of, if, for example, if you have a knife can be used to slice an apple, or a knife can be used to perform surgery, right? And although they both are cutting through something, you can see already the intended use of that knife determines the precision, the accuracy, the ensuring that there is not an adverse event. All of that comes into play. So the intended use of a device is paramount. What do you intend to do with it? So based on that, then the FDA's medical device amendment has really identified three classes, which is simple because all good things come in three, as you know. So class 1, class 2, and class 3. So class 1 is considered low risk. So what's a low risk device? Well, a tongue depressor. When you go to the doctor goes, and if a tongue depressor, that's a device, would you believe it? And and a class 1 device just needs to meet some basic criteria. After all, you don't want to put in a child's mouth or a patient or adult's mouth a tongue depressor that doesn't meet some basic criteria, right? Of making sure it's not toxic, it's not made of lead or mercury, for example. So I'm saying the class 1 has some requirements, but it's pretty basic standard. And then you go to class 2 devices and class 3 devices. And in my book chapter, that I'm so thrilled that you guys are clearly geeks like I am because you've enjoyed this chapter. The class 2, by definition, is called a moderate risk. So class 1 is low risk, and I explained examples of tongue depressors. And one of my my book editors wanted me to include in it this thing called tonometers, which are very important for traumatic brain injury and other such areas. So then class 2 devices then become moderate risk. And the language we use for a moderate risk device is you don't need approval, but you need what's called pre-market notification clearance. And the 510K is not like 401K, haha. 510K is actually a part of the regulation where it is a rule 510 with sub-bullet K, in which it actually goes into what is required. And this is the creative aspect of, because before 76, there were so many medical devices already in play. Can you imagine that in 76? So this was in before Carter got elected, right? So who was that before? I think Nixon time, actually, that's right. And who also spent a lot of money on the fight against cancer. Actually, he doesn't get enough credit for couple of good things he did. And so in that time, can you imagine, with so many medical devices already in play and companies manufacturing, they wanted to say that what if, so there were some devices that are known to be already in the market. So, and they were in the market. If they were unsafe, they were yanked out. If they were safe, they were left in. So we didn't want to retroactively go and make them file for FDA clearance. So from 76 onwards, this rule of substantial equivalence, SE as it's called, was a new regulatory construct. which said that if you can prove that you're substantially equivalent to this other device, then you are cleared, right? So from 76 onwards, as part of 510K clearance as well, all you had to do was demonstrate that if you are a new glucose meter, for example, that you have to be substantially equivalent to a glucose meter already in market. So that's a construct that was innovative, creative policy thinking. It was least burdensome to industry, so they didn't have to do an enormous big clinical studies. They just said, look, I'm making a glucose meter. There's already all of these, and I'm as good as the one that was before. They go great, right? So that was a contract of 510K. Those were the class 2 devices. And I won't go, there's, of course, all of these things have complexities in them. So I won't go into them, but I'm speedily walking through it. And class 2 510Ks, then, as Sarah briefly mentioned from the book chapter, there's something called 510Ks, special 510Ks, and abbreviated 510Ks. I'll just use an example of special 510K because you'll appreciate this. So if it's a novel glucose meter, okay, it's a novel glucose meter, it's never been in the market before. A company, let's say company Tydoc is an example of a very prominent glucose meter manufacturer. If they said, we're going to start making glucose meters from May 2021. So then they design it and they ensure that it's substantially equivalent to existing glucose meter, right? But now once that glucose meter is in place, over the next years, they can make small iterative changes. And if they make those small iterative changes, they come back to the FDA and say, we don't need to do another full 510K, but we think it's a minor iterative change, so we'd like to use a special 510K. And as a reviewer, and I was a reviewer for 6 years, and I've done hundreds of submissions, I would have to look at their claim and say, are you truly an iterative change or not? Here's an example. Let's go back to the knife or scalpel example. So if you're making a knife and you say, I want to make a small change to the knife. Okay, what is that? If it's just the holding grip, I'm going, oh, okay, you're just going from a fiberglass grip to a wooden grip, whatever, right? So I think special 510K, you're okay. But on the other hand, if your knife becomes sharper, gets serrated teeth, or has double edge, you can see it can increase the risk of that knife, right? Then I go, whoa, hold on. This knife has both sides sharp. I think this, you need to demonstrate this is not risky, doesn't counterbalance the safety effectiveness balance. So in that case, I'll say, as a reviewer, I'll say, you are not eligible for special 510K, go back and do a proper 510K. So you can see, even there, if you think about it, FDA is being creative and respectful of not being burdensome to industry, saying you can do a 510K, which is already a pretty low bar, I think. And they're saying we can even give you a sub 510K, a special 510K, if it's an iterative change. So the American regulatory system has been generally listening to industry a lot and being creative. And the last then class, so we can get off these class ladders, is the class 3 devices. These are of paramount importance. By definition, a given device or application is deemed class 3 if it's reserved for medical devices that support or sustain human life, are of substantial importance in preventing impairment of human health. So there has to be a life or death aspect to that device. Are there examples? Yes, there are examples. So some implantables, okay, could be life or death, because if it shocks, I mean, I just heard Vice President Mike Pence got a pacemaker the other day. Good luck, buddy. Let's just for a moment ponder if his pacemaker just took a shock because of voltage variation or something. So you can see it could be life or death, right? But having said that, some stents are considered 510K, which was a topic of controversy when my old boss, Commissioner Margaret Hamburg, was in office. But so some stents should not have been or should be as an argument. But you can see how some devices can be life or risk, which really plays a role in our companion diagnostic space. Now, companion diagnostics are routinely class 3 or high risk. And why is that? Because based on the results of the diagnostic, a patient who's dying of cancer right? Companion diagnostic typically is in cancer, and it also can be in other illnesses, but they have been predominantly in cancer. The person is dying of cancer, and they're going to be given a very highly effective or highly toxic drug. And don't forget, these medicines have two sides to them. They're highly effective because they only kill the cancer cell, but if you don't have that kind of cancer, they're killing the other cells. So it's highly toxic. So companion diagnostics have been generally clubbed in class 3. So that's how class 1, class 2, class 3 work. I'll take a pause here and see if Sarah wants to ask more along those lines.
No, I think you've done a great job. I do want to go back to touching on companion diagnostics, but before we talk about that, I kind of want to move forward to non-standard, specifically hitting LDTs.
Right.
Because I want to kind of pair between those two. I don't fully understand their differences.
Okay. So the way I covered this in the book chapter and is that I decided to just group them somewhat arbitrarily, but they make sense in some sense. So we saw the known, I called it known paths versus less than clear-cut paths. So the non-standard pathways are the humanitarian device exemption is very similar to the orphan drug designation that the Center for Drugs does. So these humanitarian device exemptions are only eligible, for example, for the diagnostic. It's that diagnostic test that measures a biomarker or an analyte to say if the person has Tay-Sachs disease or if the patient has Klinefelter syndrome. And I made these diseases up not knowing if they're rare or not. They sound pretty rare. They have exotic names. I don't know if they're rare or not particularly, but the rareness is defined by the number of patients diagnosed per year. So it used to be 4,000, if there are 4,000 patients diagnosed per year or below, then that is qualified for a humanitarian device exemption or humanitarian use device. That's the other term, the HUD or the HDE. Now, you would say, okay, well, so what about it? What's the big deal? This is a big deal. So the threshold for evidence to get clearance by 510K is substantially lowered. And do you want to guess why is it substantially lower? It's because, if you think about it, because they know that it's only going to be 4,000 patients or less, which has been now changed to 8,000, by the way, by the new 21st Century Cures Act. But let's say 8,000, therefore. If it's only going to affect 8,000 people or less, then that's called a contained risk. It can't, therefore the risk is known, it's a contained risk. Therefore, the threshold for demonstrating evidence is less, which is not true for diabetes. A lot of people, millions of people get diabetes. So when you're making a diabetic intervention device or a diagnostic that has a risk, that has the unknown component of who all. Classic example of these COVID vaccines. I know the J&J or J&J vaccine, for example, when they did the numbers, 1 person out of 1 million person who take the vaccine get these clots or these strokes, which is, by the way, they're saying you can get hit by lightning twice and hit by a bus twice before you can get the clot, okay? So, but you can see, nevertheless, there are millions of people who are eligible to get the vaccine. So in that case, they have had to demonstrate studies in 34 or 40,000 subjects for the vaccine approval. But now compare that to the humanitarian device exemption. The company or the whatever person is asking for it, and typically these humanitarian devices are done by a company that's making a diagnostic, and they work with the Rare Disease Foundation group. Typically a few people whose parents or loved ones have lost tragically someone in their family. They gather together to make a rare disease foundation. So the humanitarian device, they can tell you the prevalence of that, research papers and all that. Right? So the next question you'll ask is, well, then why should a diagnostic company make that? What do they get a benefit? They can only sell it to 4,000 or 8,000 people anyway. They can't make any money. Really good point. This is what companies do, and I've been a consultant where sometimes for a device, they have an intended use that can be first tested. The device's performance can be tested in an intended use that's contained. They may then make an iterative change and go to a larger market. So for example, if Sarah starts a company and she looks ready to start a company there on the blackboard and her suit on, and so she might say, I have this really cool idea that affects, that can induce in kind of energy in a person's brain who have, say, migraine, although migraine also is fairly highly prevalent. Let's find a rarer disorder. Let's say a rare disorder for which she has found a way to introduce energy into the brain by wearing a headphone or some sort of contraption. But she is a small startup company. She cannot already do a 8,000 patient study. She's going, oh, it's going to be bust. I'll never get this device out. So she says, oh, but you know what? Instead of doing migraine, I found this rarer disorder where this can also play a role, and I can help that. And so you work with the Rare Disease Foundation, and they say, yes, yes, please, thank you. Thank you for coming and seeing us. Nobody ever calls us. We leave our number on the door. So you go talk to them. You say, we can do this for you, right? And then you do that study. The device works. You get credibility in the investors market. They go, oh, wow, Sarah has an interesting device that kind of works already. Sarah, what do you want to do with this? And you say, I can make this change to do migraine. Can you give me $10 million to do a larger clinical study? So you can see that the humanitarian device not only helps rare disease patients where there's a lower threshold, but it can also be a stepping stone to making a device that's applicable to more people. So you can see intrinsically there are business interests involved in a good way, right? I mean, that's how you start something out. Right? Yeah. So that's one example of the HUD. Abbreviated is very similar to special 510K, and it's a very administrative contract, so I won't get into it right now. And then the other one that you mentioned was from my chapter was LDTs. We can go into LDTs. Oh, and de novo 510K. Oh, de novo is exciting, right? Okay. So remember, we talked about how 510K's basic construct is demonstrating substantial equivalence to something, okay? There were two, so it has to be a moderate risk, right? Therefore, it cannot be a life or death situation, moderate risk, but you have to also demonstrate substantial equivalence to an existing device. So, but what happens if you're moderate risk, but you're the first of its kind? There is no one to compare to. Then what do? Then you typically apply to the agency saying, I am a 510K. The agency says, no, you're not. They cancel you out. And then they tell you in a separate meeting, but you can come back and we'll do a de novo for you. Do we have an example of that recently? Yes, we do. The example of Adaptive Biotechnologies, which has a minimal residual disease diagnostic for multiple myeloma, is a de novo 510K. So because it's a monitoring test, it's not a companion diagnostic. So while typically one would have thought a genomic test like that would be companion diagnostic, in the case of Adaptive Biotechnologies test, it's not being used to treat a patient or deny a patient treatment. It's being used to monitor has the patient's cancer recurred or not, right? So therefore, monitoring claim is considered a class 2 moderate risk, and therefore, but then there was nobody else doing anything like that before them. Therefore, they got a de novo 510K. And the examples I mentioned in the book chapter about 2 of the TBI devices are also de novo examples as well, because nobody did them before. There again, I am not being objective. I'm a big fan of the FDA. I'm saying the FDA, which is the people's agency, has tried to be reasonable in ensuring not make it very crazily very high threshold, right? To be somewhat less burdensome, right? So that's the de novo 510K. So we covered, and the PMA is the next step. If you keep going up the ladder, the PMA is typically a, they say bad 4-letter word. PMA is a bad 3-letter word. If you are whenever Sarah starts her company, which she will after she finishes her PhD at Yale, if she goes to investors and talks about a device and they go, is it going to be a PMA? He goes, yes, it's a PMA. They're going, okay, see you later. They all walk out the door. Because why? Because PMA typically involves a remarkably large clinical study, may require hundreds of patients, has a very high threshold of evidence, because remember, it's a life or death situation risk. So the evidence threshold is very high. Now, routinely, big pharma companies and biotech companies that are in the companion diagnostic space work with a diagnostic company, and these PMAs, the clinical evidence and the data and all that, they run up to $20–25 million is what they run up to, right? And if you're doing a cancer screening test, like the companies like Exact Sciences, Thrive, and all that, they can run into $100 million because they have to, it's a average risk cancer screening test. So they have to screen as many as 10–12,000 subjects before they can identify sufficient statistically robust numbers. So you can see PMA can be a very expensive proposition. Pharma companies can afford it because, well, you know why. That's a different story. But that's the PMA ladder. That's the highest risk and cost ladder, right? But then you would say, well, why would somebody do a PMA? Well, they would do a PMA for the following two reasons. Number one, because you are working in a space where you are marketing or you want to market a device that is going to be life or death situation. Maybe it's substantially prevalent, like say lung cancer. And so you're saying, I think I can make my money back, right? That's one reason. What's the second reason? The second reason is because PMAs, you can't demonstrate substantial equivalence, you build what's called as a moat around your device. By making a given product or a given device PMA level threshold, another person who wants a copycat and come behind you, they also have to do a PMA. They cannot do a 510K because that application you're in is a PMA class 3 level application. So some companies have liked, like a Medtronic or Edwards, big fat companies with lots of money, they're going, oh, let's do PMA, let's screw the little guy, which is great, you know. So you can see there's also business interest there as well, in some sense, right? But it's driven by intended use, and it's driven by also the business strategy here. So that's where regulatory strategy, for those of you here listen to see what's the career there. Is it just writing boring paperwork? It's not. It's being able to craft an intelligent regulatory strategy. It could be, for example, pursuing a humanitarian device exemption to do a smaller population so you can go seek more money and go the bigger one, or you can do PMA or 510K. You can see there's a diversity of ways to think about it. That's why a good regulatory strategist is hard to find and does very well. Right? So I've gone over all the ladders, and I'll pause. I don't want to do all the talking, but I can go to LDT, or I can wait for your LDT question.
Sure. Maybe let's try and touch on LDT briefly, because then what I really want to get into is, and I think, thank you for mentioning kind of the role of a regulatory strategist, but I want to better understand kind of, you're working for a big pharma company. Why are you talking about CDRH, the Center for Devices? Right. Why are you not sending things through CDR, the Center for Drugs? Right. Yeah, if we could touch on maybe how often you interact with other centers.
Yeah, sure. I can come back to LDTs later when we talk about policy. So let me answer this question on CDR at CDR. Sure. So first of all, biopharma companies don't only make small molecules or large molecules. Biopharma companies also make medical devices as part of them, right? I mean, I know my company GSK used to make these asthma inhaler devices, and the asthma inhaler devices contain in them a drug that helps the patient who's suffering from asthma. So that would be a combination product, right? So, but in my role, why do I care about CDRH? Well, like I said, I work very closely with our clinical development team, as does my team, on a new drug for a lung cancer, prostate cancer, whatever it is. And as part of the regulatory strategy in it, saying, should we do it this way or that way? Should we stratify, as in take all comer population and stratify? Or should we, on the other hand, select patients going forward? I'll give you examples instead of just throwing random things. So an example in the next-gen sequencing space, from my experience with FMI is NTRK, N-T-R-K. NTRK is a biomarker. That's a pretty rare biomarker. That is, NTRK alterations are pretty rare. And two drugs that came around the same time, a year off or so, was lorlatinib from Bayer, and then there was one called entrectinib from Roche Genentech, right? And they both target the entrectinib situation. So, so in that case, the drug was targeted to patients with that alteration. So that's a very somewhat clear-cut selection. As in, would you select, would you treat all patients with entrectinib, or would you already do a clinical trial only selecting patients who are NTRK mutation positive, right? So that may be a straightforward thing, right? So in that case, it's a selection criteria. What's a stratification? Stratification is as follows. When you actually you invite all comers to the population. So let's say you work in ovarian cancer, you think you have a PARP inhibitor, and the PARP inhibitor, you think it should work in all women with ovarian cancer. But what happens, the whole reason companion diagnostics came about was this, was that in an all comers, they found when they did the statistical numbers that no, efficacy was down. It wasn't approvable enough, right? So then they said, okay, in all comers, it's not working. Is there a subpopulation that's responding? And that's how they found companion diagnostics. Therefore, they found a subpopulation in which it was highly efficacious, right? So on the other hand, if they're part of the larger group, then it won't work. It's like a Yelp review. If you only have angry people writing Yelp reviews, then that restaurant's going down. On the other hand, if you have a larger number of people who have kind of, or someone who is excited, it's something like response then, right? So you can look at it that way. So companion diagnostic in some ways were thought of rescuing drugs before. That's how it originally began. I have on my team, one of my colleagues was involved in the first approval ever of companion, was the case of Erbitux for colon cancer that Lilly and ImClone did, right? They actually did an all-comer trial for colon cancer. And the trial failed. They said, shoot, it didn't reach the numbers. So some very smart scientists in the company said, hold on, let's go back and look at these patients. Let's see if any of them, if you were to break down, they had perchance begun sequencing already patients around that time or doing PCR tests around that time. So they went back and looked, if they were to partition the patients as KRAS positive versus KRAS wild type, and I say positive, I mean mutation positive versus KRAS wild type, they saw that actually KRAS wild type patients respond great, and KRAS mutation positive don't respond to the drug at all. So in the case of Erbitux, where KRAS became the first companion, well, KRAS rescued. Otherwise, Erbitux would have never seen the light of day. While it began as a rescuer originally, going forward, drug companies are a priori designing trials taking small populations because of the fact that human genome has been sequenced. So you'll know, okay, these people, these patients have this elevated levels of biomarker versus that. So drug companies are a priori designing selection criteria. So in my role, I work with such a clinical development team, decide should we do it this way or that way. So I'm working with CDRH, I'm working with a diagnostic partner, and CDR. Because CDR and CDRH also meet all the time. So even if I go to a CDR meeting, there's a CDRH person sitting in the meeting. Therefore, the reason GSK has, in its wisdom, brought myself and I have now hired 4 or 5 people in my team is because all of these oncology trials have become combination trials, if you will, okay? So that's just an oncology area alone. But we also are, at GSK we are, and I know other companies are as well, interested in what are called digital endpoints. So let's say if you're doing a clinical trial on Parkinson's disease or arthritis, increasingly what they're using is wearable devices that actually measure mobility or things like that. So that become part of the outcome. So that would be a medical device, a wearable medical device. So there we would need to work with CDRH. So you can see the companies are increasingly becoming CDR and CDRH combinations. Long answer, sorry.
No, that was a great answer and truly informative. And I'm curious now then internally within a, not necessarily just a big pharma company, but any company, you're developing a drug or even a device, at what point does the regulatory strategist step in and actually participate in data collection or in, you know, I mean, making big decisions on go, no go with whatever is in development? Can you speak to that?
That's a great question, first of all. And it's a source of much, sometimes tension and worry, because there's always being too early or being too late situation there. And so pharma companies are evolving on this. Traditionally, a pharma company would not involve the diagnostic regulatory person early on, because like I said, there was, and I say this with love and respect, there was a contempt for, we don't need you guys, we got this, right? And that because diagnostics were as an afterthought before, right? That's how the Erbitux situation, as I explained. But pharma companies have really learned this so well now over these past 10, 15 years that they're coming early on, very much. So when do I come in, or when does my team come in? We work with the biomarker translational group within the company, work with the clinical development. And it starts at the very beginning. If you're targeting, for example, say, we just got a drug approved yesterday on endometrial carcinoma, where already there are publications a lot in the public domain that talk about association of a biomarker for a given disease, how it's highly expressed, and it may be a potential window into targeting the patient, things like that. So the medical clinical affairs group that are thinking about doing a new clinical development program are already coming in with knowledge from literature on the role of a biomarker, right? So increasingly now, from the very early on, they already start measuring biomarkers. Nowadays, right from the very beginning, my team begins to get involved into the clinical development. We attend meetings with clinical matrix team meetings, with the medicines development team meetings. And so we're beginning to be involved a lot more early on. So right from the beginning, to be honest, Sarah, going forward.
Yeah. Mm-hmm. That's truly great to know. I want to get back to those LDTs, because I kind of want to ask then, at what point, or would you ever be in a position in which you're advising someone to go the LDT route or to go through the FDA, or maybe you would go through that humanitarian device route, which you explained really well. I mean, how do you make that decision? What are some of the impacting factors? I mean, also business-wise, that are going to play into your choice there.
Right. This is actually almost odd to ask these advanced level questions at a trainee level meeting. So I congratulate you on that. These are really hot questions. So as I explained earlier, there's a cost to going to the FDA, because not only do you have to do that submission and do the study, once you are in the FDA's realm, then you also have to begin to build your laboratory up to standards, what are called as quality systems regulations, which is governed by a code of federal regulation. So that cost goes up as well, right? It's like, here's an example. I love examples. Here's, for example, if you were just brewing a bit of beer and you and your neighbor drink it, it's fine, no one cares, right? But if you are going to, before long, going, no, I'm going to sell at the local farmer's market, then the regulatory creep starts, because you don't want people to go blind with your beer, right? So, and then if you're going to about to now start canning them, bottling them, selling them at a local food market, then you really have to increase your level. So it's funny I use that, because laboratory developed tests originally were called homebrew tests, because it really was done by a local laboratory to meet the needs of the local community. So if you're living in a part of New Mexico, for example, it's very hot, people are outdoors, and the Zika virus hits, right? And But let's say that doesn't impact the Boston area, or doesn't impact colder areas, doesn't impact California where nothing lives, all the time just wind and sun. But if you are like in the middle of vegetation in New Mexico, and so the local New Mexico County medical director might say, will ask the local hospital or University of New Mexico saying, can you make a test very quick? Because I'm seeing more and more cases of people with Zika virus infections. The hospital professors say, sure, we can do that. You get smart students like yourself, and they put together a quick homebrew, if you will, right? That was the intent. So the FDA had begun to do what's called enforcement discretion, which is they said, while we have the discretion to enforce, we're going to exercise discretion and not enforce. So go meet your local needs. So in that sense, communities having lab developed tests made sense because it met local needs. Now, which is the same thinking in the humanitarian device exemption, right? As long as the risk is localized and contained, you're fine, right? Yeah. But now with large companies like LabCorp, Quest Diagnostics, and ARUP Laboratories, IQVIA, all of these people, they have labs everywhere, and they are part of one large company. I don't know if you've checked the LabCorp stock price recently. It's a pretty nice, healthy, well-fed stock price. So all these companies become large enterprises. So they began using the laboratory developed test mode. And then some of them were, in the sense that, if, for example, if you are a University of New Mexico professor and you have in your research found a way to detect Zika virus, you go, this is very nice. So the person gets tenure, associate professor, University of New Mexico Office of Technology Department, decides to file a patent on it. And then they say, well, we should make some money of this thing. So they ask a local commercial guy saying that, hey, can you produce this test? Will it be of any use? They go, sure, we can do that. So companies would say, take that, as long as they were local, fine. But before long, you can see easily, because the internet connectivity, shipping connectivity and all that, these were no longer local. They became global more and more. So laboratory developed test was, as a construct, was beginning to be abused. And things really came to a head for the following. So I don't know if you know this, at the time of Navigenics and 23andMe and these companies, I'm talking 10 years ago, more than 10 years ago now, some of these companies either don't exist or they've completely changed. I want to be careful to mention that this is historical example. So a company like, say, 23andMe or Navigenics, they were offering genetic results that will tell someone a risk for something. And they were saying, we don't need to come to the FDA because we are a laboratory developed test. And the FDA really felt that, whoa, hold on, buddy there, okay? First of all, your risk is not contained to those people in that small community. People are shipping samples from all over the world, almost, right? So therefore, you no longer, in that sense, meet. And also the FDA said, by the way, remember when I said we are exercising enforcement discretion? We didn't say we were giving up enforcement. We were saying exercising, which means they were saying, therefore, now we are choosing to enforce. And so what happened over time, Sarah, is, and so in the FDA, I was a reviewer for 4 years. And then I was an advisor to Commissioner Hamburg, Dr. Margaret Hamburg. And then I was also an advisor to Dr. Jeff Shoren, who was the CDRH office director. And I used to go with Dr. Hamburg to the hearings on the Congress, both on the House side and Senate side, because FDA has to every 5 years go and ask for money for appropriations. And that's an opportunity for all the industry to lobby Congress. This way they can do that, right? So these large laboratories, I was mentioning a couple of examples of companies that have such big stock prices, there's real money involved. This is not small money, this is real money. And therefore, they were trying to get the government to saying, you should not regulate LDTs. They should be left alone. So that was the topic of the controversy there. But going back, the foundational principles always has been intended use and risk to the patient or the population at large. So you can see the LDTs were skirting that a little bit. The LDT provision was originally intended to prevent local communities to have to go to the federal FDA agency and laboriously do a bunch of studies that may delay meeting a local community need. But you can see businesses, always smart, began to use that loophole, if you will, to try and skirt requirements to demonstrate that. And that's what you saw in the COVID testing as a classic example. COVID testing during the Trump administration, they did this emergency use authorization and all that. And a lot of them did not work, the COVID testing. Los Angeles Times published an article that said of 16 tests, only 14 of them, of 16 tests, only 2 of them worked, 14 of them did not work, right? And more recently, there's a paper published in the New England Journal of Medicine by Dr. Tim Stenzel of OIVD, my old department, and my boss, Dr. Jeff Shuren, have co-authored a paper on what went wrong on the COVID test, that the regulations were too low, right? So there's always been this regulatory tug of war of deregulation and regulation. So,
Lakshman, this is super fascinating. And speaking of failed tests and failure in general, so we have a question here in the chat from Kingsley, and he's asking, after a failed application for approval for a device drug to the FDA and a subsequent subpopulation analysis shows then substantial effectiveness or safety, is it typically that a new application is sent or required, or an addendum to the previous application is sufficient? And he's also asking, and what is the time between those two applications, or is there kind of a time frame that needs to be respected?
Well, with the caveat that specifics will always have specific constraints to it. I'll answer it in a general construct, but it may not answer specific situations. With that caveat, well, because an earlier application let's talk about a drug application. If an earlier drug application was submitted and it did not meet the efficacy endpoints statistically, then the drug application has failed. So it's like a court case, right? It's failed, but in this case, you cannot do a supplement to a failed case because a failed case was already ruled upon. But what the company comes back with is remember this failed trial number 201? We've gone back and found that there's a subpopulation with this biomarker actually do respond. And therefore, then the agency goes, oh, okay, but that's only 30% of the overall population. Your data looks interesting. You have to go and recruit more patients with this biomarker and prove your proof of concept that this truly works. So in a way, the failed trial gives them an early indication of, aha, hold on, this drug isn't a failure. It can work in some people. Then the company has to go back and recruit more patients with that biomarker positive.
Use this as the basis for a better trial, basically. So
yeah, and so they come back with a new application, right?
Okay, but then you already have a lot of preliminary data that this might.
That's right.
Okay, got it. Yeah. Thank you. This is great. I have another question. I personally have, this might be a little bit of a childish question, but how long did it take you to write this book chapter with all the tables and everything? And how long did you need to conceptualize that?
So I had left Foundation Medicine in April, and I had taken the May off, and I went 1 week. I went camping, and then so I was sitting there. It's bugs coming, so I was drinking with a couple of friends, and I would organize chapters in my head all the time, and then literally just came back, shut the door, and just kept writing. And so it took about a week to write the whole thing, and maybe about a week to conceptualize how to organize. I had to keep a flow to it because it was a book chapter. So I organized in a certain fashion. And then I had to bring in I'm not a traumatic brain injury expert. I have someone very close in my family who was in a terrible car accident because of black ice on the road. And he was in the hospital with traumatic brain injury, and he's completely recovered. He's a successful career, all that now. So the TBI became a personal story for me because I knew. So I'm not a TBI expert, so I read a lot about TBI as well. But yes, let's say 2 weeks, and maybe a little more than to clean up stuff. But yeah,
that's absolutely fascinating. I had a blast reading it, really.
Wow, that's good.
I have one more question I kind of want to throw into the mix. I know we're super tight on time, but I'm going to tie in kind of AI into here, or I guess with an eye toward what's coming to medicine in AI. Some say innovation is outpacing regulation, and I want to know what your take is on that.
Yes, and it should. It really should. I mean, how can you regulate something without knowing something completely? Like, look, I mean, Elon Musk keeps crashing his cars. So therefore, then we have to make better regulations. So, but if we did not send out these auto-driving cars and crash them, we wouldn't know what to regulate. But now that he has done that in Texas, they will have more regulation on it. So innovation should always be ahead of regulation. So otherwise, regulation will, in a way, constrict innovation. So I think in the case of AI, innovation is, I'm familiar with the work, Sarah, we were talking about at the PICC and the AI Summit, where it's digital pathology, looking at ways to be able to separate out, if you will, within quotations, biomarker positive or response positive and response negative populations. I heard another group that I spoke to recently that are not parsing out samples like that at all. They actually are parsing out, looking at a sample, they can say which subjects will demonstrate pathologic clinical response, or actually PCR, pathological clinical response. They can apparently a priori tell you, oh no, these guys will respond. And they've done that because they've seen thousands, tens of thousands of data, and they've looked at it and they have identified machine learning models. So I think the innovations here are going to be in what is the indications we have to pursue, what are our machine learning models, the extent of sensitivity. So there's a lot of that still needs to be sorted out, right? The FDA is not regulating at this time. They are listening. They are open to ideas. They're saying, no, we're here to tell us how this. But I've actually gone to the FDA on an AI/ML topic recently, where regardless of what the technologies, the core principles remain the same. What is your intended use, and how can you demonstrate performance that will lower the safety concerns but increase the effectiveness? So the core principles never change, whether it's a liquid biopsy diagnostic, a solid tissue diagnostic, or it's AI/ML. The principles are never, the same principles apply in all cases.